Good morning. Thank you for joining us today. My name is Nick Radio, the Global Product Manager for the High Content Technologies business at Thermo Fisher Scientific. The title of my presentation is New Enabling Technologies for 3D Quantitative Cell Biology Using High Content Screening. So, we have a very proud history in the high content screening space. So Solomix, now part of Thermo Fisher Scientific, invented, patented, and coined the term high content screening to reflect the needs of our early adopters that were needing more multi-parameter data than what was available by conventional technologies. So this happened back in 1996. Solomix came out of Carnegie Mellon University, commercialized the first high-content system, then called the Eureka Scan. The Eureka Scan VTI, or fifth generation, introduced a modular system that allowed for things like live cell chamber and liquid handling to be added. We had an Eureka Scan XTI with Confocal that really introduced 3D biology imaging capabilities. And then more recently, with the CX7 and CX7 laser platform, this really provided us with the means of doing high throughput confocal imaging in a way that would enable 3D research. And then just recently, here in 2020, we launched HCS Studio 4.0. I'll show more details of this later, but this provided the Eureka Scan Finder, which is a seek and find functionality that allows our researchers to find the spheroid, organoid tissue of interest and then conduct 3D imaging on it. And then finally, we have Amira. So Amira is also a new technology for us here in 2020. Amira provides best-in-class 3D analysis for the most complicated 3D biologies. And I'm happy to share some examples of that later in the presentation. So this is showing how 3D analysis began to set the stage. So basically, in 2016, there was a publication showing that 3D models were more faithfully representing what was going on in vivo. It was capable of providing more complicated assays, providing better physiological matrices, and enabling things like blood-brain barrier and other 3D environments that just aren't possible when working in a monolayer. So for the promise of more faithfully representing what's going on in vivo and hopefully being able to be better predictive of preclinical and drug safety studies, there was a significant level of investment in moving into this space. So these are just some slides that I have showing how 3D cues do influence both tumor cell growth and morphology. It's important to note that if a cell model is not in a 3D matrix, they don't have the cues, especially at the basement membrane and the environmental stimuli, to act in a way that it would in vivo. And we can see this through time course assays. We can see this through the generation of three-dimensional spheroid projections. And this is important for us because, when we look at something like oncology and cancer metastasizing, the cancer's ability to break through the ECM so that they can get into the bloodstream and potentially metastasize, this is entirely requiring a 3D environment for that type of progression. So when we're trying to evaluate ways to stop cancer and ways to stop the cell motility of cancer breaking into the bloodstream, it's important to evaluate it in these 3D matrices. And we see this, there's some pretty amazing research going on today in vivo models where we can track this. We can show differences in cell shape, differences in cell speed in a 3D environment like you see on the right-hand side versus what happens with HL60 cells in a monolayer, where we're not seeing the same level of motility, we're not seeing the same level of progression, and even the cell morphology, the basic cell shape, is different than what you see in 3D. So that leads us to the Cell Insight CX7 laser. This is our latest high-content screening platform that was really designed to facilitate confocal-based screening. So it gets its name because it's a 7-color laser-based system. The lasers are important so that we're able to provide not only high-quality imaging, but also high-throughput confocal screening. So in a drug discovery environment, faster is better so that we can figure out of a different chemical library which of those compounds has positive therapeutic effects. The system was also designed for versatility, so it provides both software and laser-based autofocusing to enable anything from flat bottom plates to U-bottom plates to hanging droplet plates are compatible with the system. And then I think one of the most important things for our customers is its ability to do on-the-fly phenotyping. And what this means is that without any reservations or asterisks, any bioapplication that you're running on the CX7 laser, you're doing imaging and analysis in real time. So this is just showing some of the positive impacts that having a laser has on imaging. So this is just showing 2 images of the same exact fluorophore specimen. So these are cells that it's a whole cell stain and then Alexafluor 514. On the left-hand side, this was imaged with our CX7 LED platform. On the right-hand side, this is the same but with the CX7 laser. And basically, based on the physics of the flatness of a laser compared to a light-emitting diode, you are going to see reduced vignetting, better image flatness, which does provide for superior contrast, better resolution, better level of detail, with significantly lower background compared to what is present in an LED-based system. The second positive advantage that's really important, especially in 3D models, is the ability to visualize deeper into a tissue. So similar to the last screen, here you're seeing 2 spheroid image sets. On the left, this is with the CX7 LED, and on the right, the CX7 laser. Now, it's important to note that these spheroid samples were approximately 300 microns in diameter. And what you can see as you go deep inside of the spheroid, in the LED-based system, and I actually am going to bring up my cursor here so I can show you this, but we see as we go deep into the spheroid shadowing, and that shadowing is essentially the LED light is not bright enough to penetrate the deeper areas of the spheroid, which leads to the shadowing effect. And the deeper you go into the spheroid, the more prominent that becomes. Now, if you look at a similar sample with the CX7 laser, you note right away that shadowing effect is no longer there. And the reason for that, again, it's just physics. We have a higher powered light source in an LED or a laser compared to the LED. Based upon that higher intensity laser, we're able to illuminate the structure more evenly, which allows us to visualize all the cells from the periphery down deep into the core. So if understanding what's going on deep into the structure is important, then utilizing a laser compared to an LED is one of the advantages that you're going to want to take. And then lastly, as this last image is showing, when we have a laser-based system compared to an LED, and we're talking about doing 3-dimensional imaging, where we're doing not only multiple colors, but now multiple Z slices in each of those colors, there's a true economy to scale on what that does on a high throughput screening time. So this is just showing a comparison of a laser over an LED, and you can see there's approximately a 5- to 6-fold improvement in throughput time when you are doing a 20-slice, 70-micron confocal going across 4 different colors. And the more wells that you're screening, the higher that advantage is going to be, just as we're getting more and more images being compared. So here, these are CX7 laser-derived confocal screens of A594 spheroid cells that were screened on the CX7 laser, again, just demonstrating the advantages to high-throughput screening. So in those instances where there are a lot of compounds or concentrations or drug libraries that need to be evaluated, a laser-based system will provide you those answers in a shorter period of time. And then finally on this screen, I just want to show when we're working through a 3D environment. So again, it's not just the X and Y, but now we're going up into the Z. So here you're going to see a video. What this is showing is each individual Z slice as we're going from the bottom to the top of this neuronal 3D spheroid model. And with that, we're able to go through and make the quantifications. And now you can see it's going toward the bottom. And with that, this will set us up for how are we going to analyze the spheroids. One of the things that has been a challenge, especially in flat bottom plates, is spheroids have a tendency of being relatively randomized across the bottom of the plate. So you can see that here in this schematic. Unless you're using something like a U-bottom plate or a hanging droplet plate, there's really no easy way of controlling where the spheroids are going to locate at the bottom of the well. So in order to provide a solution to that problem, we have introduced Eureka Scan. So Eureka Scan is a seek and find functionality where it essentially does a double pass. At the first screen, it's going to do a lower magnification, so in this schematic a 4x, providing a larger field of view. And what this is going to allow us to do is to identify where the spheroids are at. It's important to note that with Eureka Scan, you can apply this to any bioapplication in HCS Studio. And because of the real-time analysis capabilities, as we are imaging in that low mag, we can also validate in or out the spheroids that we want to quantify. So in this case, in the spheroid example, you can see that this spheroid was invalidated based on size, shape, intensities. And then in the second scan, at a higher magnification, in this case 10x, we're able to only image those two validated spheroids that were not gated out. Now this is a 3D talk, but I also want to mention that you can also apply this for tissue or rare events. If there's a specific phenotype or a specific event that you're interested in quantifying phenotypically, you can apply Eureka Scan for that application as well. And to just kind of bring it home, this is showing a application of Eureka Scan with before and after Eureka Scan is applied. So this first slide, which was provided from us by Dr. Chi Yan, in this case an important collaborator at a New York University School of Medicine. So what Dr. Yan and colleagues provided was, so here is a spheroid plate. It was seated in a 96 well flat bottom plate. There were one spheroid per well seated in the top half of the plate, so you can see one spheroid. And then in the lower half, rows E through H, there are two spheroids per well. All right, so when we're trying to take a higher magnification image of a spheroid, that is going to require, in a flat bottom plate, 25 fields. Again, because the spheroid can be present anywhere within the bottom of the well. So here you can see all 25 fields, and the problem is the spheroid at 10x is only going to take up one of those fields, one of those images, meaning that 24 are wasted. There's no data. It's an empty field. And you can see that across the top half of the plate, so A2, A3. Here at the bottom, again, there are two spheroids in the bottom half of the plate. So at most, you're talking maybe 3 fields of view, like in this situation in F2, where you're actually having to take images. Again, the majority of the fields have nothing in them. So because the spheroids can go anywhere in the bottom of the well, you're forced to take all 25 fields. And what that does is it really increases your scan time. So here you can see it takes a little over 28 minutes to conduct this 10x screen using the 25 fields. Now with Eureka Scan applied, so here you can see instead of doing a 10x image, we're doing a 2x image. So 2x low magnification. This 2x scan, now it's only one field per well, during which we're able to identify the XYZ coordinates of where that spheroid is located. This took 40 seconds to go through and screen because we're taking one field per well, not 25. And now, during the second round, we can share the XYZ coordinates to the higher mag, and now you're able to see that only one field instead of 25 is taken. So we're significantly cutting out the wasted images. We're able to get the higher resolution images necessary to see the spheroids closer up. And what that means for our actual comparison is that you're going to see two distinct advantages. First, for the scan time, without Eureka Scan, it's a little over 28 minutes, as we said. When we apply Eureka Scan, both the low mag and high mag together, that took a little under 2 minutes. So there's about a 14-fold savings in throughput time with Eureka Scan applied. And then because we're not taking so many empty images, there's about a 15-fold improvement in image file size. So when we're doing the 25 fields, it took us 2.8 gigabytes. With Eureka Scan applied, they cut it down to 187 megabytes. So a full 15-fold improvement in savings. Now, usually people are not wanting to do this in a wide field mode. They're wanting to do it in a confocal mode. And with that, instead of doing one Z-plane, they're wanting to do multiples. And this is really where you see a fairly significant improvement. So here you can see just a comparison with wide field imaging and then the same spheroid, but with 11-slice confocal ZStack, all generating our ability to get much better resolution, removing the out-of-plane imaging so that we can see the spheroids inside of the cell. And that is going to provide us the ability for quantitative analysis. So I just wanted to share what that actually means when we look at this. We can do a first pass, low mag to do where the XYZ coordinates are provided, and then do the second pass at 10x with laser autofocus applied, 11 confocal slices. And when we do that, we can do the confocal screening and still have it completed in under 7 minutes. If you recall, it took 28 minutes just for a wide field screen with no confocal, no Z slices. So a significant improvement in performance and throughput. And that's where we're trying to emphasize here that Eureka Scan will allow our customers to do multiple ZStack confocal screening for their high throughput assays by improving the efficiencies of where it is that we're doing our higher magnification screening. And I should point out, Eureka Scan is unique in that you can apply up to 3 consecutively higher magnification scans. So if you wanted to go even above 10x, you could do that as well for even higher resolution imaging. Okay, so now I want to kind of introduce the second new technology that was introduced for the Thermo Fisher high content platform, and that was Amira. So Amira is a 3D analysis technology that was pioneered by our partners at FEI. We've spent the last 9 months working pretty closely with them to customize Amira for 3D analysis off of the Cell Insight platform, primarily the CX7 and CX7 laser. So I want to provide some examples of what that means. Amira provides best-in-class 3D and 4D analysis. Anything from basic assays like 3-dimensional cell counting, segmentation, to much more sophisticated assays that just aren't possible on other platforms. things like 3D morphology, things like 3D neurite tracing, things like 3D co-localization for IO, immuno-oncology, can be conducted with the Amira platform. So I want to provide just a couple of visuals to hopefully provide some examples of how this can be applied in your own lab. So here, this is just the ZStack Confocal. Here you can see that we're doing three-dimensional cell counting and segmentation off of the image set. And it's important to note, before I move forward, it's important to note that, as I said, Amira has been customized to recognize the cell inside images, meaning that our customers will see a streamlined performance. The ZStack images coming off of the cell inside can be directly imported into Amira, and that will allow our customers to begin their 3D analysis right away in a streamlined fashion. So why is important? That's demonstrated here in these next two slides. What you're looking at, these are spheroid images that were acquired with the CX7 laser. On the left-hand side, you are seeing the tumor in a untreated state. So these are negative control wells. The left-hand side is DAPI. The right-hand side is a dead cell dye marker. The images on the right, let me bring the cursor up again. This was treated with 1,000 nanomolar paclitaxel. And pay attention to this treatment compared to this treatment. You can see with the paclitaxel, deep inside of the spheroid, we see a significant increase in the dead cell dye label, indicating that, as we would hope, paclitaxel is able to induce cytotoxicity deep inside of that spheroid. I think it's important to note that on the untreated cells, you're really only seeing dead cell death at the periphery of the spheroid, whereas the cells deep inside of the core are nice and healthy, unfortunately. So this is one application. We can also do this with things like organ on a chip. We can do this with things like Fluidigm or Lumentis. So here's another example. These are ZStack images taken with a 70-micron confocal pinhole setting. They were done on an organ on a chip application. So this is just showing a visual of those images. And not only are we able to show the three-dimensional visualization to get an idea of what this three-dimensional spheroid projection looks like, we can also generate our quantitative measurements. So things like cell counting, volume, how circular or sphericity of the objects can be all quantified off of that 3D measurement. So if you need to find more than a 2D projection and you need to see what's going on deep inside of that organoid or spheroid or organ on a chip, these are all applications that are now enabled with the Amira software. And then finally, I wanted to leave off of a zebrafish assay. So this is done where bright field imaging combined with confocal RFP CX7 laser imaging. You can see in red the liver organ that is being identified, and let me zoom in on these. So here at higher magnification, you can see three-dimensional reconstructions of the liver, again what we see here quantified. And then with Amira, we're able to showcase our customers what's going on with the organ. We can show them how circular it is. We can show them the overall volume inside. So here, in this case, so A1 is an untreated well. H6 was with a compound known to induce fetal alcohol syndrome. And you can see a fairly significant reduction in the volume of the liver compared to the untreated control. So these are some of the new capabilities that are inherent with Amira when we're doing 3D analysis using the CX7 and CX7 laser platform. I'm confident that our users, when they combine Eureka Scan to more efficiently find the sample of interest with the 3D analysis capabilities of Amira, will really help to accelerate this next generation modeling. And the last thing that I just wanted to point out is in the Amira FEI site, our users will be able to access protocols and recipes so that if they want to do a three-dimensional neuroid outgrowth measurement or a 3D immuno-oncology colocalization measurement, those protocols will be readily available. The only thing that the user would have to do is validate that their serial number to show that they're actually a customer, and from there they would have access to the portal. And with that, I think we're right on time. I want to leave you, these are really nice CF7 laser confocal Z-stacking images. I think it's important, especially in the oncology space, to note that this really is what is unique about three-dimensional imaging and analysis, the ability to see what's going on inside of the spheroid at the cellular and subcellular level. And quite frankly, we're just excited to partner up with some amazing researchers out there and hopefully be part of the solution for next-generation oncology treatments, next-generation autism treatments, next-generation developmental neurotoxicity treatments. And with that, I really appreciate your attention. Signing off. Thank you very much.